let's now look at some examples of exogenous hormone use and gland atrophy. I say check out the last homework packet question, the challenge question at the end, as well as patient A scenario in the review slides that I had posted. So let's start with the homework packet first. All right. This is the last page of the homework packet. I have it listed as page 29. Let's blow this up a little bit. And you're given a table with information, numbers here associated with the glands, organs, and we have to know what those numbers mean. And so you always want to check what the units are here. And we're being told that these organs are being weighed in milligrams. So these are organ weights in milligrams, right? A common misconception people have when they first see this type of diagram is they think hormone levels, because that's the way we've been talking about this information, you know, blood plasma concentrations of hormones, but we're not looking at that here. We're looking at the weight of the organ itself to see whether it is overproducing given hormones or it's underproducing hormones. So we're looking for gland hypertrophy versus gland atrophy to give us an idea of the hormone levels without actually measuring them specifically. And this question states that rats were injected with hormone X, and we're comparing those weights for the rats that were injected with hormone X with control rats to see whether we've had hypertrophy or atrophy or no change between those two treatments here for our rat organs. And the question itself wants to explain why hormone X is CRH. So it's telling you what the hormone is, and to look at the results and explain, justify why that is the exogenous hormone that is being injected in this case. And that's something else we need to keep in mind, is that this is actually hormone injection and not a disorder that we're looking at here. Where the CRH is being upregulated because we have an oversecreting tumor, for example. That's not the scenario. The scenario is we're injecting a certain hormone. What is that hormone? And I might ask you in this way, maybe as a multiple choice question. I might say, how do you justify that this is hormone X? Or I might ask you to determine what the hormone is. So I might not tell you that hormone X is CRH, but I might give you similar data table here and ask you, what is hormone X? Is it TRH? Is it, let's list them, TRH, TSH, TH, or is it associated with the adrenal axis, right? Is it CRH, ACTH, cortisol? It's just some examples. So these might be your choices. Okay. So I might reframe the question in that way. So let's try to answer it in that way. Let's say that we did not know that hormone X was CRH, and see if we can walk through the information here to figure out that it is indeed CRH, right? And where I would start here is with any of the values here for the rats that were injected with the hormone, and whether those values haven't changed compared to the control, so that we can rule out some of these potential hormones, right? If we look here at the thyroid gland, the rats that were injected with hormone X, they have the same thyroid gland weight as the control rats. There was no change in the thyroid gland. So that suggests that whatever hormone we were injecting is not influencing the HPT axis in any way, right? Not influencing the levels of thyroid hormone in the rat circulation. So we can actually rule out all three of these here. It's not concerning the thyroid gland, the thyroid axis. So that leaves us with the adrenal glands. So let's go to the adrenal glands and the pituitary. What we're not given here is the hypothalamus and say the weight of the hypothalamus, right? We would have to infer that, for instance. So we see here that the adrenal glands for the rat that's being injected with the hormone, those adrenal glands are larger. So these are hypertrophied adrenal glands, right? They are enlarged. And what that suggests in terms of hormone injections is that we are not injecting cortisol. Because if we were injecting cortisol, we would be replacing the function of the adrenal glands, replacing the function of those zona fasciculata cells. They wouldn't need to function, they would shut down and they would atrophy. If we are replacing a given hormone or a given gland, then we should see gland atrophy. If we see hypertrophy instead, that means that we're not replacing that given hormone, but we are triggering that gland. Right? So this can't be cortisol that's being injected because we're not getting gland atrophy of the adrenal glands. Then we look to the pituitary because we don't have to look at the thyroid. And pituitary, with respect here, that would be ACTH and as a potential hormone that we're injecting. Compared to the control, right, those rats that we're injecting with the unknown hormone, well, those rats have a much larger pituitary, right? Which would suggest that those corticotroph cells that are releasing ACTH are actually enlarged and overproducing ACTH. We also have hypertrophy here. If we were injecting ACTH, well, those cells in the anterior pituitary that produce and release ACTH, they can shut down. They don't have to do their job. Right? And we would see atrophy instead. But since we see hypertrophy, we know it can't be ACTH. And thus we are using the power of, deduction at this point to suggest that it must be the hypothalamus in that job that we're replacing here with the hormone injection. It must be injecting CRH. And with those high levels of CRH that we are injecting, right, the exogenous CRH, then those neuroendocrine cells are atrophied. If we were able to weigh the hypothalamus, we would see that, that the weight would be lower there, which is what we're inferring with this answer, that CRH. And we should see the same pattern if we think of the actual axis that we're affecting now. So we have ACTH that then affects the release of cortisol. If we're injecting that exogenous CRH, then we are going to see high levels of CRH in circulation. We would measure high levels in circulation. And those high levels of CRH would be overstimulating the pituitary cells, causing hypertrophy of those cells, and overproduction of ACTH, which would then overstimulate the zona fasciculata, leading to overproduction of cortisol. But then if we circle back within the endogenous pathway with the high levels of cortisol in circulation, we see we have inhibition of those hypothalamic cells. Right? They're overpopulated with cortisol, those receptors, and those neuroendocrine cells in the hypothalamus that produce and release CRH, they are inhibited. They shut down their production of CRH and they atrophy. So here we would have atrophy occurring because we have this negative feedback inhibition. And so if we could parse out the endogenous CRH and the exogenous CRH, we would see that endogenous levels are low, while exogenous levels are high because they're actually being injected, put into the body. So I mentioned I had a question similar to this in the scenarios that I had in that study guide. Let's find it here. Endocrine system, my hybrid review. We had looked at B and C for the HPT axis. Now this is scenario A. What slide is this? This is slide 8. And here we're saying we have an individual, this is a post-mortem exam, so we can actually weigh their glands, their organs. So we can see whether glands and organs have atrophied or hypertrophied. And we can also measure the hormone levels within the blood as well, circulating, what would have been circulating levels. We do that for CRH and ACTH and the cortisol. So let's look at what that axis is telling us in terms of the information. CRH, our ACTH, and then our cortisol. We find that this individual has low levels of CRH, high levels of ACTH, and high levels of cortisol, right? And we see that the pituitary gland is atrophied. The adrenal cortex is hypertrophied. So we have enlarged adrenal cortex, adrenal gland. Put an H here for hypertrophied. And our pituitary gland, however, has atrophied. All right. Now we didn't weigh the hypothalamus here to get information by hypertrophy or atrophy, but we don't even need to go that far because we already see a disconnect here. We see that we have hypertrophied adrenal gland and we have high levels of cortisol. That makes sense, right? But then we look at the pituitary gland and we see that it is shrunk, it's atrophied, but the levels that we see within circulation of ACTH are abnormally high. Right? And there's only one way that we would have atrophy of a gland and high levels of that hormone that would supposedly be produced by that gland, and that is the scenario where we are injecting that hormone. Right? So what we would infer from this finding right here that doesn't seem to match up is that this individual was taking exogenous hormone injections of ACTH, right? Hormone replacement. And we don't even need to go to level of CRH here, because really for CRH, we would predict that these levels should be low because of the high levels of cortisol. Right? That would be a normal response by the hypothalamus. But the normal response by the pituitary to the low CRH should be low ACTH, and it's not. We have the high levels, which would suggest that it's actually being put there. So that's a signature that you would be looking for if we were looking at, you know, a scenario that's not rats being injected, but an actual person, and asking, are they undergoing hormone replacement therapy? We're looking for that disconnect between the levels that we're measuring in the blood for the hormone and the size of the gland organ that's producing that hormone. If we see high levels of a hormone, but gland atrophy for the gland that produces and releases that hormone, those two factors combined tell us that that hormone is being injected. It's exogenous. Right? Does that make sense? Okay. So definitely expect a question or two like the homework packet question, similar to this type of question as well. Right? Always have that in the back of your mind when you're looking at the levels and you're told something about atrophy and hypertrophy of glands, that maybe you're looking for hormone replacement in that type of scenario. And then you have to narrow down what hormone is being replaced. Okay.